area of my research that I found particularly interesting, which was looking at how diversification of African forms has led to the current distribution of restricted endemics in Northeast Africa. So I'm going to focus on that primarily for the next half an hour, and then hopefully we'll have a bit of time at the end and I might be able to go through some of the more wider continental relationships I've looked at. Okay, so I have to admit I'm coming into this very much from a molecular background. I am a molecular biologist, and I'm going to give you a bit of a background to the genus, but I am well aware that the majority of you will probably know more about this family of birds than I do. Okay, so I have been looking at the genus Zostrops. They reside within the family Zostropidae, and this genus is currently the largest genus within the family. It encompasses 70% of all the recognised species. Now, members of this genus are remarkably uniform in their phenotype. So their structural morphology, their plumage coloration, their plumage patterns, and their behaviour is all very similar. And this has made designation of taxon boundaries within the group really difficult. And as a result of that, this group has quite an unstable taxonomy, which has caused a lot of problems, especially for me. Okay, so whilst many of the forms are generally sedentary in nature, so they don't move around a lot, the group's best known for its exceptional colonisation abilities. So they're well known to colonise an island system and then speciate there. Okay, so we have got this conundrum with the fact that they are quite sedentary, but at the same time they have these fantastic colonisation abilities. And what has happened with that is these colonisation abilities has given them a massive distribution. So they occur in the Afrotropics, the Indo-Malay zone, and also in the Australasian ecozone. Now, this exceptional colonisation ability has led to them being described as a great speciator. And because of this, there's been a considerable amount of research effort that has looked at insular systems. But whilst there's been quite a lot of work that's done on the relationships and the colonisation abilities and speciation patterns of island taxa, the relationships of continental forms, specifically mainland African forms and Indian forms, were largely ignored. And that's kind of where my PhD started from. So, concentrating on the African system, just to clarify, throughout my PhD I've adhered to the taxonomy of Dickinson, 2003. So if it's a little bit different from what you know, I apologise. But according to Dickinson, continental Africa and its associated island systems. Now, by associated island systems, I mean the Gulf of Guinea, the Indian Ocean, and the Gulf of Aden. So that entire system encompasses 14 Zostrop species and 4 Spirops species. Spirops being another genus within the family Zostropidae. So within mainland Africa, what we have is the majority of Africa is occupied, so this yellow area here, is occupied by 14 subspecies of Zostrop senegalensis, commonly known as the yellow white-eye. Okay, so there are 14 described subspecies, and they occupy a diverse range of habitat from acacia woodland to evergreen forest. Okay, so very widely distributed subspecies. And then as you move over to the lowlands of the dry northeast, the yellow white-eye is replaced by the Abyssinian white-eye, or Zostrop abyssinicus, so that's the bird down here at the bottom. There are currently 6 subspecies currently within Zostrop abyssinicus, and they only occur within lowland habitat, so they are adapted to lowland type environments. Now, this species also occurs outside of mainland Africa. It occurs on the island of Socotra in the Gulf of Aden and also in the Yemen. Now, much of southern Africa, down here, is occupied by Zostrop pallidus, or the Cape white-eye. Now, this species has had a considerable amount of taxonomic instability. Previously, there have been up to 3 species recognised in this area, based primarily on variation in belly coloration. Belly colour. You do get quite a bit of variability in southern Africa. But the most complex area, and the area that I'm going to talk about today, is in northeast Africa, where we have 8 subspecies of Zostrop polyagaster, or the montane white-eye. Now, this species occurs from the mountains of Ethiopia through the Kenyan highlands to several isolated mountains in southern Kenya and northern Tanzania. Now, this species is generally much larger than the other African forms, and it tends to have a much richer plumage colorisation. So it's got a much richer green back, and it can generally have a white or a grey belly. Now, some of them, they have an expanse of the white eye ring, so that can get quite large, and sometimes you find this crest of golden feathers on the forehead. So these birds, these montane birds, are very typical of this montane habitat in northeast Africa, and they're ecologically segregated from the neighbouring species Senegalensis and Abyssinicus. Now, previously, because of the presence of Highland Senegalensis, so within this East African region, you have Highland Polygaster and Highland Senegalensis, the difference being one of them integrates and the other one doesn't. Now, previously, because of the presence of these Highland Senegalensis, all these non-integrating montane populations were treated as conspecific with Senegalensis. So later, an alternative treatment was adopted whereby the non-integrating montane populations were treated as subspecies of a wider species complex under the name Polygaster. So that just gives you a bit of background on kind of what's happened with these populations with regards to the taxonomy. Now, this is important how they're currently grouped at the moment, because the fact that they are taxonomically represented as subspecies of a wider species complex means that we're presuming that they're relics of a previously widespread population. So if they do all belong to the same species, then they would be relics of a previously widespread population that are isolated on these montane forest fragments. So in order to understand the current distribution of these endemic montane forms in northeast Africa, I had to start looking at what's happened in East Africa over the last few million years. And in doing that, I start looking at the climatic record and the geological record. So just for those who might not be aware, over the 5 million years, there's been a general trend for an increase in aridity and a decrease in temperature towards the present. And this general drying trend has been punctuated by periods of extreme moisture, followed by subsequent periods of extreme aridity. So what you've got essentially is these wet and dry cycles that have been happening over the last 5 million years. Now, these wet and dry cycles are thought to have had a profound effect on the vegetation associated with this area. Now, in these mountain systems, the vegetation at different elevations will vary in its susceptibility to climate change. So when you have these periods of pronounced aridity, what would happen is in these lowland environments, you would have a change from a forested area to a savanna scrubland type habitat, or in some cases, a semi-desert type habitat. Whereas during periods of increased moisture availability, it would turn again into a forested habitat. So what you've got over the last 5 million years is an expansion and a contraction of forested habitats up and down these mountain slopes. Now, this contraction and expansion is thought to have had a profound effect on the diversity of birds within this region. And the current taxonomic treatment works against a model called the montane speciation model. Now, under this model, there would have been a previously widespread population during a period of moisture where it was connected, and it occurred in the lowlands and the highlands. When it became more arid, these populations, as the forested area would have contracted up into these high elevations, what would have happened is these forest birds would have tracked this suitable habitat up to higher elevations. Once there, they would have become isolated on different mountain peaks. Now, once isolated, they'd be geographically separated, and over time, they would diverge to become subspecies of a wider species complex. Now, that is what, until now, we have thought has been going on. Okay, but this hasn't been tested. So we've put together a taxonomic framework based on something that we haven't tested yet. And what I was looking at is, okay, yeah, this model, it does explain the current distribution of Zostrop polygaster, but it's not the only model. It's not the only way we can get these restricted endemics on these montane peaks. And another model I started looking at was called the gradient speciation model. Now, under this model, populations speciate along an gradient. Okay, so what you would have is during periods of increased aridity, so when it was getting more drier, the climatic tolerances of the habitats would get narrower, and this would cause is a habitat gradient. And what you would end up having is you would have a population, and one of them would adapt to a forest habitat, and the other one would adapt to a savanna habitat. What you'd end up, essentially, two closely related species, but one of them would be a forest specialist, and one of them would be a savanna specialist. So I started looking at these two models and trying to look at the differences between them. Whilst they can both explain the presence of montane endemics, they both imply very different roles for natural selection. So under the current taxonomy, and according to this montane speciation model, these endemic montane populations conform to niche conservatism. They're not able to adapt to new or changing environmental conditions, and therefore these montane populations are relics, and these montane forests are functioning as a sink. Okay, so these ancestral populations have no ability to adapt to new or changing environmental conditions. In contrast, the gradient speciation model says completely the opposite. It's saying that these ancestral populations could adapt to new and changing environmental conditions, and these climatic fluctuations would have promoted divergence within this region. Now, an intermediate of these two models is something called the vanishing refuge. Under this model, smaller mountaintops would, in times of prolonged aridity, so when it was dry for a very long time, they could become unstable. And what would have happened is that these forest fragments would have become too small to maintain a viable population. And under this model, what would happen is that once the population became too small, it would promote species to adapt to less favourable habitats. So they're not adapting straight away. Only when they're losing their habitat do they adapt to this habitat that, you know, it isn't their first choice, but they'll go there if they have to. Now, these two are both based on ancestral populations being adaptive, but under this latter model, the population has to go through a severe bottleneck, so the population has to become really small, and then with a subsequent expansion. So I ended up now looking at these three models and saying, okay, well, we think we know what's going on. We haven't tested this. So I've looked at the different models. Let's see what the genetic data says to us. So what I was looking at, really, I was looking to accept or reject the montane speciation model. Now, in the literature, if you read about the montane speciation model, there are three main predictions. And the first one is that lowland taxa should be ancient relative to highland taxa. Okay, so if these mountain populations, they've ended up isolated on these peaks, they aren't adaptive, they're relics, they shouldn't then be able to adapt to lowland environments. So you're saying that anything that is in the lowlands should therefore be ancient relative to these highland populations. It's also saying that speciation events, they should coincide with periods of climatic instability. So if you're saying that the climate has affected these birds, you should be able to match it up with a period in time, with the climatic record, and say, okay, at this time the climate was very variable, and this is matching up with the divergence events. Okay, and this third one is that you should have these stable montane areas will have promoted differentiation of population, leading to a collection of restricted endemic taxa. What that essentially means is that all of these isolated populations should be closely related. They should form a single clade, so a single group in a phylogeny. Okay? So, I'm not going to spend a huge amount of time going through all the molecular methods because I don't want to bore everybody to death. But if anybody is interested in that and wants to come and speak to me about it afterwards, I'd be happy to go through it. I'm just going to point out a few key points that I think are really important. And the first one is looking at sampling. Now, when I went into this project, I had no idea how hard it would be to collect samples for birds. Now, the samples just for this small region, I was looking at the whole of Africa. My PhD was to tackle the whole of Africa. And I thought, oh, I'm only going to tackle a small section, it should be fine. But the stuff I used in the study has actually taken over a decade to collect. So there's been various research groups that have been collecting in East Africa for several years, and I begged, borrowed and stole as many samples as I could to try and get the most comprehensive sampling as possible in this region. And because of this sampling effort, I managed to get exceptional coverage of these montane endemics. So I managed to get 5 of the 8 subspecies in total. I also managed to get the 2 highland senegalensis subspecies. I got samples for those. And I also got the two jubensis, so that's the white-breasted white-eye, from Kenya and Tanzania. So I managed to get two of those subspecies. We also got sampling from outside of East Africa, so a bit more sampling from outside of Africa. We've got quite extensive coverage of the Indian Ocean and the Gulf of Guinea. Also some samples from India and also from Australia. So this was an absolutely massive sampling effort. Now for markers, I wanted to look at nuclear genes and mitochondrial genes, and I tackled both of them using 2 mitochondrial genes and 1 nuclear gene. Now unfortunately, when I went to sequence the nuclear gene, there was no sequence variability. Because there was no differences between the sequences, I couldn't use it. It was essentially not informative. It wasn't useful in this context. So all the results I'll be showing you today will be based solely on mitochondrial sequence data. Okay, so the analysis, I only want to say a couple of things about this. The first thing that I have to do is look at the relationships between them. And to do this, I have to construct a phylogeny, which essentially, I always describe it as a family tree. It just tells you how things are related. The next thing I did was to do divergence estimates. And what I was doing is trying to date of the time that these birds diverge from one another. Once I had them divergence estimates, I could then look at the geological record, I could look at the climatic record, and see what was going on during the time that these birds were speciating. Okay, so this is my East African phylogeny. This is generated from a concatenated data set from 2 mitochondrial genes. We have cytochrome B and ND3. Now, I realise this is far too small for anybody to read, so please don't try and attempt it. What I want you to concentrate on is the colours, and I'll talk you through the colours and which species they belong to, and you should be able to follow the key points of what I'm trying to extract from it. So the first point is, this was a massive achievement for anybody who works on white eyes, because this was the first strongly supported molecular phylogeny with comprehensive sampling of mainland white eyes. Okay, so this was a huge step forward. So the results showed us that of the African white eyes sampled, mainland African white eyes sampled, they fall out into 2 major clades. We have Africa A here at the top, and we have Africa B here at the bottom. Now both of these clades contain oceanic island radiations from the island systems on either side of Africa. So if you look in Africa A here, we have one radiation from the Gulf of Guinea and another one from the Indian Ocean. There is another group, which is from the Indian Ocean. That's the ancient Indian Ocean white eyes, and they're actually coming out at the base of the African phylogeny, which isn't something that's new, that's something that has been shown in other phylogenies. So if we look at the colours represent the different species, so the current taxonomic treatment. So the red represents the montane white eye, so that's the East African montane white eye. Yellow represents the yellow white eye, so the widely dispersed white eye. Blue represents Zostrop pallidus, so the Cape white eye from southern Africa, and green represents Zostrop abyssinicus, or the white-breasted white eye. Now, the first thing that I hope that all of you should be able to see from that is that these colours aren't grouping together. They're coming out in completely different places in the tree. And what that tells us is that they're all invalid species. So essentially, we had 4 species. We thought we had 4 species. We thought we understood them. But what's actually happening is that they're all non-monophyletic, which essentially means that they're not forming a single clade. Now, what I would have expected, according to the montane speciation model, is that all of these red blocks, which are all subspecies of Zostrop polygaster, they would all come together to form a single clade. And what's actually happening is they're actually more closely related to other taxa, which are completely divergent in their habitat, their ecology, their distribution abilities. So when I first was presented with this, it was a good job I didn't know that much about white eyes, because I had to completely bend my thinking because it just wasn't adhering to the current taxonomy. So as I've said here, there's extensive non-monophyly of all mainland African taxa, specifically within Zostrop polygaster and Zostrop senegalensis, because that was most representative of my sampling. Now, although there is extensive non-monophyly for the species, so maybe unsurprisingly, maybe surprisingly, there is very strong support for the monophyly of subspecies. So what we've got here is, although Zostrop polygaster, so the montane white eye, is not supported as a species, all these individual subspecies have strong support. Now, these stars here, what they represent is very strong support. So strong support are greater than 95%. And what we've actually got in most of these cases is they've actually got the maximum support. So coming away from this, I was thinking, well, actually, maybe these individual endemic populations actually represent independent species, and that they're not subspecies of a wider species complex. Now, the second thing that's really important about this data set is actually when we look at these montane white-eyes and we look at what they're closely related to, it's taxa that occur in different areas. The best example of this is if we look at this clade here. Okay, so this is a subspecies called mubalensis. It occurs in the Chulu Hills of Kenya. Now, at the top of this, you have this subspecies. It's endemic to this one region. In the lowlands, you have the white-breasted white-eye. Okay, only occurs in the lowlands. You wouldn't have ever have put these two together. Nobody has put them together. But actually, when you look at it, they're sister taxa. So that's quite interesting. You also find it down here, but down here it's a bit more complex because here you've got two montane subspecies. Now these is Culalensis and Kikuensis. These occur on Mount Kalal and Mount Kenya in North Africa, I would say. Now those two peaks are actually relatively close together. But what we're seeing is that those two aren't closely related. Actually, this Kikuensis that occurs on Mount Kenya and on Aberdare's range is more closely related to the nominate subspecies of Senegalensis that was taken from Ghana. So we've got a bird that's occurring on a mountain in Mount Kenya, and actually it's more closely related to the nominate subspecies, two samples taken from Ghana. Now this clade here contains Zostrops senegalensis jacksoni, which is a highland senegalensis from Kenya. Okay, and then we've got the nominate subspecies from Ghana. What we have here, Berlini, which is the highland senegalensis from Tanzania. So I was very confused by this because I thought, okay, we have highland senegalensis throughout this region, and I thought they would come together in a group, but they haven't. They've actually come out in two completely different clades. So I hope you can agree with me. This is a very, very confusing group. So going back to my predictions. I was looking at my montane speciation model predictions, and I was saying, okay, lowland taxa should be ancient relative to highland taxa. That is not the case. If we go back to this phylogeny here, these are lowland taxa, these are highland taxa, and in no way are these basal to these highland taxa. Okay, so that means that we can reject this first prediction. Now, we can't answer this second one yet because I haven't done my divergence estimates, but the second one, they should form aggregates of restricted endemic taxa. They're not forming a monophyletic clade, so we can reject that as well. So everything that we have based our taxonomy on so far, well, it's not working out as we thought it would. So here we have some divergence estimates. Again, please don't try and read the phylogeny. It's massive, and you just won't. So look at the colours. What we have here is we have different nodes, and we have bars on these nodes. These nodes are basically error bars. And you will see that they are quite wide there. Now, what this is, I did two different types of analysis. Now, the first one is based on an avian molecular clock. I'm not sure how many of you are familiar with the avian molecular clock, but basically that assumes it's a global clock for birds and says that they all evolve against a clock. Okay? It's highly unrealistic, but if you don't have calibrations based on fossils or geological events, then it's the best thing that you can use. So that's one approach that I decided to use. The second approach that I decided to use was to use a calibration. Now, as far as I'm aware, there is no geological record for Zostrops, so I used a geological date of an island to calibrate my phylogeny. Now, anyone who knows anything about dating will notice straight away I've only got one calibration point, okay, which does make it weaker, and that is why I have these large error bars. But what I can tell you is that from these two different approaches, they did bring out very different dates, but what both of them told us is that the majority of divergence events that occurred within this group have occurred during the Pliopleistocene, during these climatic fluctuations. Now, these blue bars here that are occurring, these these bars indicate major global climate transitions. Okay, so they are associated with major periods of glaciation. Now, whilst that wouldn't have encroached onto Africa, what that would have done is it would have affected the intertropical convergence zone, which would have affected how Africa was affected by climate variability. Okay, you wouldn't have had ice there, but it essentially, during the blue periods here, it would have been very wet. In between these blue bars, it would have been very arid. If you look at the mean values, okay, so forget the error bars for the moment, but if you look at the mean values, divergence of mainland taxa coincides with these dry period. So the nodes that are looking at these divergence between the mainland taxa occur between these dry periods. Now, what happened during these periods of dry periods? The forest was fragmented. Okay. Now, during the wet periods, this is when you have colonisation of the surrounding island systems. Now, I realise that there are very wide error bars on that, so I am pushing it a little bit to state that as fact. But I do think that's a very interesting fact anyway. Now, what we do have is the divergence of all these montane endemics, according to the calibrated approach, all happened within the last 0.88 million years. So this makes this group a very recently diverged group. They are very recent radiation, which is interesting in itself. And when I first went into this, I thought that divergence events between these montane endemics would have occurred during this period of climatic variability. I had no idea that all the divergence events would happen between this period of climatic instability. So essentially, when I did this analysis, I was very surprised because actually this period of climatic change might have not only affected divergence of the montane endemics, it actually may have had a massive effect on all African white-eyes. Okay, so it may have had a bigger impact than we previously thought. So we've gone back to the montane speciation model. We've already said that two of them we can reject, but one of them we can accept. Okay, but we can't accept the montane speciation model, so we looked at the other models. Okay, so we'll look at the gradient speciation model and the vanishing refuge model. Now, at the moment, I cannot distinguish between those two models because to do that, I would need to do population analysis because I need to test for a bottleneck. So to distinguish between the vanishing refuge model and the gradient speciation model, I would need to see whether those populations previously had been reduced to the point that it would cause them to diverge. Okay, so that's the only way I could do it. So I can't statistically say the difference between them. But what I can say is that in this group, when we're looking at these montane white-eyes, what we have noticed is that divergence in this clade and divergence in this clade, it's actually matched up with periods of pronounced aridity. So the more arid it becomes, that's when you're getting this divergence between this widely distributed species that also occurs in savanna-type habitats. Okay, so again, it's a bit of a stretch for me to say, but that is something that I need to further look into. We need to go and look into the population genetics of it and see what's going on there. Okay, so the key points from my work, just this chapter really, is that this phylogeny, what this made me do, and hopefully will make other people do, is challenges the utility of morphological markers within this family or within this genus. Most of the current taxonomy is based on morphology and distribution, and what I've shown within this East African group is within this group it can't be trusted. Okay, so we've really got to think with this group exactly how we are going to categorise them and how we are going to group them. Also identifies possibly a significant underestimation of zooxanthellae diversity within mainland Africa. If those montane endemics are actually all independent taxonomic units, straight away that could possibly be a new eight species. So as I was moving on from this, I was realising actually it may be a lot more complex than I previously thought. As it says here, a much more complex evolutionary history than I thought it would be. I previously, going into this, thought that what I would be looking at is species that trapped forest habitat and became stuck. And essentially, it's a lot more complex than that. I've rejected the montane speciation model, and most importantly, what I think is most importantly, is I've identified that these ancestral populations are actually adaptive. Now, this is important because it means that these montane forests, they're not functioning as speciation sinks. It's not a case of, these birds can't go anywhere. They do have the ability to adapt to new and changing environmental conditions. And I do think that that's an important point to make. And it's an important thing to think about when we're making kind of conservation decisions on how we are treating these montane forest habitats. So, I'd just like to thank a few people. This is Dr. Julie Day at University College London and Dr. Robert Preece-Jones. Thank you very much. For fieldwork, I'd like to thank Nigel Clear, who was absolutely fantastic. I have to admit, I've admitted to a few people, I'd never touched a bird before my PhD. I was a little bit terrified of them, to be honest. And Nigel came with me and was brilliant. He taught me how to take blood samples and how to mist net, and it was one of the best experiences of my life. So I really do need to thank him for that. I'd like to thank the National Museums of Kenya, who were brilliant in trying to organise the trip with me and take me to the sampling localities. They were fantastic. I'd like to thank all of these people here for their samples, their various samples. This phylogeny would not have been possible. And I really think it's important to stress the importance of working together and collaborating, because to do these big phylogenies, it is not possible by yourself. It's really important for groups to collaborate together, to share their resource, to try and find out these difficult questions. Finally, I'd like to thank everybody who's given me money, and I'd like to thank you for listening. If anybody has any questions.